Model organisms are an invaluable tool for research. They tell us a lot about our own biology.
Model organisms like Drosophila or C. elegans or zebrafish are so valuable because their signaling pathways are highly conserved.
The advantage is clear. We can study questions that have broad implications for human health in much smaller and less complex models.
So typically you have to inject, you have to breed, you have to screen and characterize to find the right line for your purpose. The downside, generating those transgenic lines is simply a tedious process.
Screening and sorting is usually done under a stereo microscope because you want to go quickly through your dish and you want to pick the right organism for downstream analysis.
It would be great to use stereo microscopes to characterize those transgenic lines, but you face simply the problem of the autofocus blur, especially in 3D specimens.
And if you try to capture this as an image, then you have the autofocus blur and the information in one plane.
The blur disturbs this image quality so much that you cannot use it simply for the characterization.
With the THUNDER Imager Model Organism, we can now address questions we never thought about.
Whether it's brain, Drosophila, Z. elegans or zebrafish, with the THUNDER Imager you get crystal clear images of three-dimensional specimen thanks to computational clearing.
Now we can see through the haze